everyone. Thank you so much for coming. So we have today, this is our InnovATEBIO PCR company showcase today. And following this, we'll have a 3-part series coming up. The program for today is that right about 3:15, or as soon as we get done with this introduction, we'll have Carolina Biological Supply Company, and then followed by that would be the MiniPCR, and then Biorad and BABIC in that order. The program is that we have about 15 minutes for each company allocated, and then which includes an introduction to the products that they have, followed by a short Q&A. I believe you have a flyer, and you can, I have the permission to say that you're welcome to email the presenters with any questions following this showcase. Again, as you know, we are recording this webinar, this session. It will be available on InnovATEBIO website next week. And once we are done with the 4-part presentations by about 4:15, I believe, we do have some time allotted for any further question answers that anyone has. So with that, I believe we're ready to get started. I'd like to call on Dhani Biscocho from Carolina Biological Supply Company. I hope I did not butcher your last name, Dhani.
You did not.
Great. Dhani is the Product Manager of Biotechnology, Forensics, and Genetics at Carolina Biological Supply Company. Dhani, the call is yours.
Thanks, Purnima. Good afternoon, everyone. Thanks to everyone for being here today. And thank you to InnovATEBIO for inviting me to chat about some of our PCR products and resources that are available to you. Like Purnima mentioned, I'm Product Manager. My name is Dhani. I work at Carolina Biological. Do you guys see my slide deck up right now, or you can? Okay, good to know. All right, so this morning I decided that I wanted to start my talk off a little different. So we're going to work backwards in my slide deck. So it's a Flip It Friday type of thing. So I'm going to start by showing you guys the free resources that are available to everyone here. So because I think that's going to be important to you, and you don't have to sign up for anything. You don't need any special passwords or be an existing customer even. You can go there right now if you wanted to consume all the resources. Actually, if you would like, please visit the website, and if you want to follow along with me, because if you guys forget about some of the stuff I talked about today, remember to go here, because we kind of allocated all these fun resources and videos and assets for you guys to use. So let me stop sharing this, and I will share. Sorry, 1 second here, guys. All right. And I'm going to reshare my other screen. Okay, do you guys see this? It's carolina.com/knowledge. Does everybody see my screen? Okay, yeah. Great. So this is our landing page for all of our digital assets and all of our articles and all the content that we write for instructors and educators. If you go to Life Science, there's a drop-down menu. And then if you go to biotechnology, this is the biotechnology landing page. So on this page, you're going to see the most recently featured article or piece of content that we have. And it's written by us or our partners. You guys can get everything from step-by-step procedural videos, ideas on how to start a biotech curriculum. So let's go into this one, for instance. This is our most recent series of articles that we're coming out with this month. It's called Biotechnology Labs Pitfalls and How to Avoid Them. And it's a very simple article that basically outlines, what these molecular techniques do, a couple of things that to watch out for when you're performing them. So for instance, here's bacterial transformation. do pick the right size colonies to make sure that they're in proper growth phase. Make sure that when you're doing your heat shock, have your water bath set at appropriate temperatures. It even comes with a video that outlines the entire process from beginning to end. It continues on with electrophoresis do's and don'ts, which will be important for you guys if you're performing a PCR in your lab, because that's the final step to confirm if your PCR worked or not. A couple of do's and don'ts here as well. How to use controls. We even have this handy dandy guide that you can download a high-res version. If your gel doesn't look like the perfect gel, we're always in search of that perfect gel. If your gel doesn't look like what it should look in the teacher's manual, here are a couple of guides to tell you, hey, maybe you overloaded the DNA, or maybe there's not enough DNA, or perhaps you didn't dilute the buffer properly. So here are a smattering of incorrect gels along with an ideal gel to kind of like walk you through the process and make sure that you've made your gel properly. And of course, we have more procedural videos and PCR chain reactions, polymerase chain reactions, do's and don'ts as well. So using your controls and whatnot. So that's just one article of many articles that we've compiled for you guys to use together. I was talking about procedural videos. If you are, starting off teaching these biotech techniques, we have a really nice video that discusses how to use a micropipette from beginning to end. Maybe I can play a little bit of it and just let it play. Okay, so here, this is how you pipette, and it goes over everything that you need to know.
The pipette shows the
when you are teaching how to pipette. So it discusses how to set particular volumes, how to find the first and second stop to expel like that little piece of liquid that you have at the very end of your tip. And we have many other videos amongst like a lot of these articles, how to pour a gel, how to run a gel, how to properly stain and destain a gel. So please kick around some of these articles and kick around on this website, even bookmark it, because we would love for you guys to use this. And like I said, it's a free resource. One article that would be important to you would be this buying guide that we put together for our PCR kits. If you go onto our website, it's very quick that you might be overwhelmed by the selections and different permutations of kits that we have. So we put together this, which PCR kit is right for you buying guide. So for instance, we have a kit that deals with the bitter tasting, the SNP, the PTC. If you go to this buying guide, it gives you like a quick synopsis of what this kit is about, the type of sample used, the type of stains that are available with the kit. Here it has a little prompt, best for teaching. So this is a good kit that you would use to show the relationship between a genotype and a phenotype, and the importance of SNPs. And this goes for all of our kits. So we have about a dozen PCR kits, and we have the synopsis for each one of those. And if you wanted to, you could, oh, I'm interested in this kit. If you wanted to click on that, it's hyperlinked, so it goes straight onto the landing page for that kit. And you could do a little bit more research and get a little more detailed information about these products. Okay, so let me unshare this. So please, carolina.com/knowledge, that's the landing page for all of these biotech resources. A ton of videos, a ton of articles, information about our kits, curriculum resources. Please check it out. I think even if you're a veteran, I think you'll find something useful in there. Okay, let me go back to my presentation here.
That's really nice.
Excellent, Ed. Yeah, we think so. It's like we have all these great resources, and sometimes they're just strewn about on the web page, and it's difficult to have like a repository for all those things to live. And like this is a very easy, pleasing to the eye way that you can find all these things. So yeah, I'm glad that we got to put that together. Okay, let me continue on with my presentation here. Let me share my screen. Okay, so we have a diverse kit offering. We started with, PCR of lambda DNA many years ago. Back in the late 90s, we were one of the first companies to supply PCR kits to the educational market, and we've really grown and had a lot of offerings, and we have about a dozen or so kits, and we cover topics like barcoding, bioinformatics, forensic scenarios, GMO foods, epigenetics, and a lot of those genotype to phenotype connections that I think your students would really like to see. So our PCR materials, we use PCR beads for our PCR reactions. They've worked very well for us in the past. It gives you very consistent results. You can leave them at room temperature for over a year, in my experience. And it's really convenient because there's less pipetting, maybe less pipetting error. All you need is your template DNA and your primers, and you're set to go. We also offer proprietary stains. We have our Carolina blue stain, which is an Enithidium bromide-free option that may work for one of your classrooms or your labs. Yeah, we really like to pick some of the best materials from different places to put together in a kit, and we do the hard work of optimizing everything together so you get very good, reproducible results every time. With that, you get exclusive content, exclusive protocols. We've worked with the DNA Learning Center at Cold Spring Harbor for almost 30 years now, and we brought a lot of these biotech kits to the education market, and they're not only are there researchers there, they're educators there. So these protocols have been vetted very thoroughly so that these things will work in your classroom. That being said, we support you guys all the way. If you guys have any questions about the kits, if you purchased one of our kits and you didn't get the type of result, like I showed you some of those pictures of those not so perfect gels, if you have one of those gels, take a picture of them, send it to us. If you are thinking about buying a kit, contact us. Maybe you'll be talking to me and I'll walk you, step by step from beginning to end to see if that's a good fit for you. But if you're having challenges or results that are, kind of in between, inconclusive, not what the teacher's manual says it should be, just drop us a line. I mean, we're happy to help you guys, and pictures definitely help. So we're not going to leave you guys hanging. So yeah, we're here if you need us. And you're going to be talking to a real person, and we usually get back with you within the day. I'm close to my time, so maybe I kind of jogged through that a little bit, but I wanted to give you guys a chance to ask questions. This is my contact. Email me, call me anytime that you guys have questions. InnovATEBIO has been really great with us to reach out to like a new audience, and we want to serve you guys because I think you guys are doing really important work and training the new workforce.
And I'd like to welcome Allison Nishitani. Allison is a curriculum specialist at MiniPCR Bio. Allison, we are ready for you.
Am I appropriately sharing my screen?
Yes, you are. We can see it.
Okay, perfect. Too many monitors. Thank you so much for having me. My name is Allison, and I do work on the curriculum team at MiniPCR Bio. Before that, I taught high school biology for several years, and before that, I was a molecular biologist. Today, I'm going to share just a tiny bit about the resources from MiniPCR Bio that are designed to help you get PCR into the classroom. MiniPCR's overarching goal is to make biotechnology more accessible. One side of our work is that we make portable biotech equipment like the MiniPCR thermocycler. One great aspect of the MiniPCR thermocycler is that they enable scientists and their students to perform molecular biology outside of a traditional lab space. Here are some photos from researchers and their students using MiniPCRs to study frogs in the field in the Amazon, and my personal favorite example is that the MiniPCR is also used by astronauts on the ISS. But MiniPCRs are also great for students in the classroom because each group gets to program and monitor their own reactions. The MiniPCR app is really intuitive to use. Students can program it faster than me, even though I've been doing PCR probably longer than they've been alive, because it looks like an app. You can do it on your phone or on your computer. Of course, the app shows the basic information for the PCR program, but it also has features that are designed specifically for student engagement. You can track the temperature as it cycles in real time, see animations of what's happening in the reaction at the molecular level to help students make the connection that during the extension step, the polymerase is actually copying the DNA, for example, is the step that's shown here, and track the estimated number of copies of the target sequence. All of these features are designed to help students better understand how PCR works at the molecular level. It's not just some black box where they put their samples in, and then when it's done, it's done. They can actually track it and better understand how it works. The other side of MiniPCR's work to make biotech more accessible is that we offer innovative lab activities to give students real hands-on experience with biotech in the classroom. Because we believe that hands-on experimentation enriches student learning, we have an extensive catalog of labs that use molecular biology to dive into core concepts in the study of biology. Today, I'll give you a super brief overview of just a few of our PCR-based classroom activities so you can see the breadth. We truly believe that anyone can do molecular biology and have lots of resources to support first-time users. If your students are new to biotech methods, we have great resources including written explanations, videos, and student worksheets that explain core techniques like micropipetting, PCR, and gel electrophoresis. And to help make PCR experiments as accessible as possible, we have some labs that come with prepared template DNA. These are great for students new to PCR because it means you only have to pipette 3 things: the template, the master mix, and the primer mix. The topics that are covered in these PCR labs that come with prepared template are appropriate for students of all levels, from general high school through college, and I'll give you 2 examples right now very quickly. One example is the Missy Baker Missing Lab, which is amazing because it's so versatile. The lab is set up as a missing persons investigation, and students test DNA from 2 suspects to see if it matches the missing Missy Baker. Obviously, one way to use the lab is to talk about forensics and using DNA to identify people, but because Missy Baker has cystic fibrosis, and that's how students identify her in the lab, you could also use this lab to talk about genetic disease and the relationship between genotype and phenotype. The lab comes with great visual questions that walk students through the molecular cause of cystic fibrosis. And depending on whether you want to talk a lot about cystic fibrosis or just focus on the forensic side of things, this lab is appropriate for general high school or advanced high school students. Another lab that comes with prepared template DNA is the antibiotic resistance lab. This lab uses a case study to introduce a very pressing issue, the prevalence of antibiotic-resistant bacteria in the environment. In the lab, antibiotic resistance is said to have been discovered and tracked to a specific farm, and students are testing soil samples from neighboring farms to see if the resistance has spread. The lab can be adapted to be appropriate for students of all levels. In particular, I really like this lab for advanced students because examining antibiotic resistance is a really good way to talk about evolution. We also have several PCR labs where students extract DNA. We have the classic PCR experiments that everyone loves, like using PTC to link genotype and phenotype, or testing foods for GMOs. But today I'll quickly highlight 2 of our more unique offerings. First, I'm a big fan of our plant genetics lab because it links Mendelian inheritance of a trait with the underlying molecular biology. In this lab, students genotype fast plants for the gene that controls the stem color phenotype. I also like this lab because it's applicable to almost any level. For general high school, you could focus on the link between genotype and phenotype, And for a more advanced class, you can get into the specifics of the mutation and the recessive allele and learn all about transposable element insertions in plants. The last lab I'll tell you about is our sleep lab, which is unique because it focuses on a genetic association that's currently being studied in humans. So it's kind of like an open investigation. In this lab, students genotype themselves for a locus that's implicated in circadian rhythm phenotype. This lab is great for advanced high school or college level classes and gives students a chance to investigate the genetics of a complex trait, like whether you have morning preference or evening preference. I want to point out that all of the MiniPCR labs comes with complete lab curriculum that you can download for free. You don't have to buy the lab to access the materials. Each lab comes with background information, easy to follow lab protocols, and pre and post lab questions. And as a former teacher, I really appreciate that each lab also comes with editable PowerPoint slides to make it easy for you to customize the presentations to your students. To find the entire lab curriculum for any lab, when you're looking at a lab on our webpage, you'll see a tab that says Downloads. It's not the first tab selected. If you hit the Downloads tab, then you'll see all of the free downloads, the lab guide, the classroom slides, and any other free resources that are specific to this lab. We also have full demos of most of our labs available. This gives you a chance to see the labs in action and also shows one way that you could introduce the material to your students. If you had to go remote again, you could even have your students watch the webinars and then interpret the data that's presented there. I only had time to tell you the tiniest bit about 4 of our PCR labs, but I'll point out that we have an extensive selection, including a new PCR genotyping experiment that's a follow-up to our CRISPR-Cas lab where students knock out a gene in bacteria. We also have a soon-to-be-released lab where students will actually collect soil samples and test the environmental DNA for antibiotic resistance genes, and a COVID qPCR lab where students will test simulated patient samples for SARS-CoV-2 infection. So if you're interested in receiving updates about these new labs, you can scan this QR code, which will sign you up for a mailing list. I'm happy to answer any questions now, or you can email me directly at any time. My email is allison@minipcr.me directly, or you can also use the last link that I put in. Anyone can sign up for a one-on-one session with one of our scientists and curriculum designers, where you can ask us questions about the equipment, about the curriculum. We can really help you find the best solution for your individual needs.
Pretty amazing. I want to know more about that sleep.
The sleep lab was one of my favorites. Actually, Sebastian, my colleague in the chat, is the one who wrote that lab. He studied circadian biology for his PhD. It's a very cool lab.
Yeah, fantastic. I see there's new message. Any other messages?
In the CRISPR
it is
lab,
part of it. Sebastian feels it is part of 23andMe panel. I'll have to go look it up. That would be a great thing for you guys to say, or you should tell 23andMe about what you're doing. They might support it.
To answer your question, Lori, in the knockout lab, they are doing a gene knockout. So it's not a targeted mutation in the bacteria. The Bio-Rad lab models that. In our lab, it's just a straight gene knockout.
Great, thanks. Zoom, the challenges of Zoom. So hello, everyone. My name is Lee Brown. I know some of you, and some of you I know your names, and some of you I haven't seen before. So I work for Bio-Rad Laboratories. I'm a curriculum and training specialist for them. I've been there for now 11 years, which is hard to believe. And I'm just down the road from Linnea and the rest of the crew at ACC. I'm in New Braunfels, Texas, but I cover really a third of the country. So there's only 3 people who do what I do. So I'm going to just quickly show you our kits and our thermal cyclers as well. And, we can talk, if you have any questions or anything. We also have a whole seminars coming up with Innovate as well. And I'll put that flyer up too. So, whoops, there we go. Oh, hold on. Yeah, let's start here. Oh no. You know what? let me go. I hit the wrong button. There we go. Okay, so I wanted to just quickly start off with a lab that is not a PCR lab, it's a simulated PCR lab, just because it's relevant to COVID. And so this is one where we're giving your students quote unquote pre-amplified samples from patients. It's based on an actual case study that came out of China early in the pandemic back in February, where these restaurant goers were, one of them got sick, and then gradually more and more of them got sick. And so they started testing the patients for COVID. So this actually walks your students through the gel electrophoresis of those, quote unquote pre-amplified samples. Obviously, they're not real human or COVID samples, so no worries there. And then they have to figure out how might COVID spread. Okay, so we're kind of having them look at where the air conditioner is, who served who, what tables are close together, and they're coming up with different hypotheses to see how COVID spreads in the classroom. This is really designed for teachers that want to introduce the topic of PCR but don't have a thermocycler. It's something that's relevant. It's pretty inexpensive, and you can do gel electrophoresis with it. So that's just kind of a way to ease into things. If you're looking for something for a general biology class, for example, might be great if you don't need to actually teach the hands-on PCR. For PCR kits, we have just a traditional crime scene and suspects. This is CSI kit. We also have that kit, sorry, let me go back. We also have that kit available as a real-time kit where you can use that on a qPCR, real-time PCR system if you have one available. I'll talk about ours in a minute. That same real-time kit we've modified to simulate COVID testing. So this would be something that is appropriate for a molecular biology or a biotechnology techniques course where you are actually doing real-time PCR. Again, they're not human samples, but they're labeled as different patients. And you can see here the samples on the right and what your output should look like on the left. Okay, so a good way to teach the concepts of real-time PCR, teach serial dilutions, which is one of the best parts about teaching real-time PCR, I think, is seeing how good their pipetting skills are. And it allows you to kind of talk about all the testing that we're doing now. I think this is COVID testing is suddenly everyone knows a lot about COVID testing and PCR, right? So this is a good opportunity to show students actually how that's done using simulated samples. We have PV92. I know some of you are doing this. This is for students who are extracting DNA from their cheek cells and looking at allo repeats. So just a very straightforward homozygous or heterozygous, look at where they are. They can do some Hardy-Weinberg. So that's a pretty traditional one. We've expanded our barcoding offerings. We used to just have fish. Now we have it for mammal, insect, and fungus samples as well. So my nickname for this kit is now like the Crunch, Fluff, and Puff kit, but they haven't, marketing is not on board with it for some reason. So anyway, it's our DNA barcoding kits. You can get it as all of those or as just the fish one. I'm sorry, this price isn't right on here. This is just for the fish. I'll have to look up the price for all of them. And we have our GMO kit, which allows you to bring in samples from different foods. The samples that students bring in, we encourage them to use, different produce or Tostitos chips, organic and natural, or organic and regular. Hawaiian papaya is always genetically modified. So we have a whole list of things. I use soy chicken nuggets a lot, the Morningstar Farms ones. So I like to use those. These give really good results. My favorite part about this lab is it's very well controlled. There's positive and negative controls and plant and GMO primers for everything. So they can see if they were successful, not only with DNA extraction, but also with amplification of their PCR samples. And if that GMO gene is present or GMO sequence is present, okay? So looking at our thermocyclers, Bio-Rad, I think everyone's probably familiar with us, but we've been around since the 60s, forever ago. The RAD actually stands for not awesome like you would think, but it's for radiation. So back when, that was cool in the 60s, but now we just get to call it RAD. So we've been doing PCR since the 80s, and this is just kind of a timeline. I don't want to go through the whole thing of it here, of course, but what our last year has been spent doing, 2020 and 2021, is a ton of COVID research, and our clinical diagnostics lab has been putting out a lot of testing as well. So I've got our offerings kind of bucketed into just conventional PCR, endpoint PCR, right? It's that first line, real-time PCR or qPCR, and droplet digital PCR. We're gonna have a whole seminar in droplet digital, so I don't wanna get too far afield in that. I'm not an expert in droplet digital. I know, this much of all that. So, but I will introduce it to you guys. So our endpoint thermal cyclers are here. This T100 on the end is in, I don't know, hundreds of thousands of labs, I feel like, all across the country, in university research labs, clinical diagnostics labs, high schools, community colleges. So this is just a workhorse. This is the one I use in all my workshops as well. 96 wells. These guys over here are just if you needed to run, this is 2 blocks of 48. Same for this. We also have 384 wells. This guy in particular, the C1000, you can add a real-time component on top of that. So that is one that you can actually expand out into a real-time system if you want. Okay. So the T100 is, just ask me for a quote if you want one. Typically around $24.95. Sometimes we have promos. Touchscreen. I've never once had to train a student. Teachers always want me to train them on this, and I'm like, just have your students look at it and they will immediately figure it out. It's very, very simple to use. And it's pretty quick. Cycles are anywhere from, I think we've gotten down to 90 minutes up to 3 hours. It just depends on what you're amplifying, right? So our real-time systems, we just came out with these guys on the left, these green ones. They're the Opus series, and those are our new real-time series. The one on the far right, this is what that real-time system looks like if you swap out that C1000. If you swap it off with an optical head on it, then you get that CFX 96 real-time system. Okay, the CFX Connect is one that you will need a computer to run. The others you do not. Okay, well, you need a computer to see the output. Let me say that. Okay. So those CFX real-time systems all come with the CFX software, and it is, I don't have my own CFX, so I don't get to do this a lot, but whenever I go into schools, I just have so much fun playing with all this because it's really, really powerful, and the analysis is pretty deep. So you can actually play with this online on our website too, if you wanted to look at the software, or train your students ahead of time. Droplet digital. So again, like I said, we're going to have a whole seminar on this. I'm just going to give you the quick and dirty rundown. Droplet digital is actually an endpoint PCR, and it enables you to look at individual molecules of DNA. Okay, so if you're looking at your reaction, typically when you're doing it in a PCR tube, you have a mixture of template DNA and non-template DNA, right? All swirling around in there, and you're just amplifying the template DNA. So endpoint conventional PCR kind of gives you a yes or a no answer. If you wanted to do quantitative, you really needed to go to real time. Droplet digital gives you a way to do kind of both of those. So you take your initial sample, you generate droplets from them. So these are our droplet generators. This guy is a manual one. This guy's an automatic one. And those droplets are created, they create 20,000 droplets, okay, per well. That's then put into a conventional PCR system. So this is just our CFX 384, I think that's what this one is, or I'm sorry, not our real time, it's our C1000, okay? So PCR happens in each of those little wells, in each of those droplets independently, okay? So these drops you're seeing here, the green ones are positive, where'd my cursor go? And the blackish ones are negative, right? And then you have to read that out. And so this system right here is what is used to read those individual droplets of whether they're negative or positive. Then you can see, okay, well, I had this percentage of positive droplets in my sample. It allows you to quantitate your template, right? So you don't need a standard curve for this like you would in real time, because you're just quantitating it just within each sample, okay? So let me just show you a couple more screens just to kind of get your bearings here. So this is what that might look like, right? I have my sample in here, all mixed together, run it through the digital droplet. It makes these oil and water droplets, very teeny tiny. And then each droplet may have one or just a few molecules of DNA. They might be negative, they might be positive, there might be a mix. We're not going to have a ton of samples per droplet. And then your droplet reader will read that out and tell you if it's positive or negative, okay, or if that sample's there or not. So again, endpoint PCR, you don't have many indeterminate results. It's either yes or no, right? You don't need to create a standard curve. And the really cool thing about it, especially with the automated droplet generator, is from user to user, it's very standardized. So you don't have that user variability there, okay? Here's kind of what it would look like if you had, you know, no targets, low targets, all the way up to high targets. Okay, and this will be, again, per reaction law. The systems here, there's, you know, I don't even have a price because there's so many different options and ways you can configure these. But this is your reader back here. This is a droplet generator here. This guy's all in one. And we have these 4 clinical diagnostics labs as well. These are the research use only ones. We're using this, I think there's probably 5,000 papers at this point. That there was 4,000 just last year. So it could be more than that at this point. But we're using these a lot in environmental monitoring. The webinar that we'll have will be talking about wastewater testing, wastewater monitoring for COVID. And so you can get an idea of how that works and how they can tell what concentrations are in the wastewater and, you know, kind of identify how prevalent COVID is in a community, like in a college dorm or whatever, okay? Liquid biopsy, CAR T cell therapy, CAR T therapy, these are all things that are just kind of exploding in the world of droplet digital right now. Our advanced PCR series is here. I think you guys have all got a flyer. What I'm gonna do real quickly is put this in the chat. There's a whole list of resources. And I just wanted to show you a few of those real quick, and then I will be done and I can answer any questions. Here we go. So on our classroom resources link, can you guys see this okay? Okay, perfect, thank you. There is a whole section on here for PCR, and this has our playlist, it has animations to show your students, PowerPoint slides, it has real-time webinars if, you know, if you wanted to show those or learn yourself. I would not recommend just letting a 1-hour real-time seminar run for your students. That will be a little bit mind-numbing. So, I mean, it's interesting stuff, but, you know, we also have, let me move this menu bar, our playlist. These have all of those biotechnology techniques. I always think everyone knows about this, and every time I put it up, people are like, what? I had no idea. So there's all these things on how to pour gels, how to streak plates, how to do all the labs, PBLO and different PCR labs. And we just added all these little, you know, Sharpie whiteboard, you know, I'm sure you guys have seen them like in commercials and stuff. So this is where the little guy with the Sharpie's drawing them out. They're real quick. They're like a minute. Gives your students a real good lab overview of the lab itself. Droplet digital PCR, if you're looking to find out more about that system, this is just 5 minutes and explains it really well.
To decode.
Whoops. I think that's it. That was a lot. Hopefully I got it all in, but I'll stop sharing and you're welcome to ask me questions if you have any.
Thank you, Lee. That was fantastic.
Yeah, sure.
I have to admit, I have one question, and is there any way to find out how many wastewater labs have ordered that droplet PCR machine, or the distribution of the droplet PCR machine between all your customers? It'd be interesting for our students and faculty to know that, because we're preparing the people to work in these labs.
I'm sure Sharon will have more information. I don't know off the top of my head, but I know it's been extremely prevalent this year and last year. Yeah, it's been, there's papers coming out, like, it seems like once a week we feel, you know, we're getting more and more of those. So yeah, pretty common.
So obviously I need to invite environmental science faculty to all of these seminars.
That would be really good. Yeah, I would think that yes, Lori says it's very expensive. It is. It's up there. So I don't, it would be good for them to know about for sure, because I do think this is kind of where the industry is going. It allows, it kind of bridges the gap between real time and sequencing. A lot of people are just replacing next-gen sequencing with doing digital droplet to check for things, because it's much faster and less expensive than sequencing for sure.
Yeah. Well, I hope everyone doesn't mind I bring this up, because now I've started to do metagenomics, which is next-gen sequencing, which is looking at environmental samples.
Yeah.
So another way to look at environmental samples.
And you can do this with whole cell applications as well. So yeah, there's a lot. There's a lot there that like I said, I only know about this much. So I'm hoping to learn more from Sharon as well. So
great.
All right. Great. So our last part for today's session is I'd like to invite Ying-Soo Lo. She's the Executive Director of Bay Area Bioscience Education Community. Ying-Soo, you're up. Thank you so much.
And
hi,
thank
everyone.
you, Ying-Soo.
Yeah. Hi, everyone. I think I know most of you on this call. So maybe I don't need to do my spiel. Well, anyway, it's happy Friday. It's crazy here in the Bay Area because we've got Fleet Week going on. We've got the Dodgers against the 49ers in baseball. So it's very happening place. And I'm sorry you're not here. BayBec is also having a workshop tomorrow as well. So I am Ying-Soo Lo. I'm the Executive Director of Bay Area Bioscience Education Community. And we are located here in the San Francisco Bay Area. And right now we have our lab located in Burlingame. We also have a location in City College Mission, but we don't have access to it right now. But in any case, we are busy at work in the lab and helping high school classrooms at this time. So I'm just going to share my screen and tell you a little bit about BayBec to start off, and sort of something that we do. And I know this is a PCR. This is a PCR showcase, right? So I will get into that, but I just want to start off with showing you what BayBec is all about. And I titled this, we build biotech awareness in the high school classroom. If you're a community college instructor, or even if you are a high school instructor, we really try to build in this pathway to community college, four-year school, and beyond, like, what's his name in Toy Story? I always forget. Buzz Lightyear, him. So, and how do we do this? So let me tell you a little bit about us first. We are about 25 years old, and we do four main things, four of our services. We have teacher professional development, and just like Biorad, what we do is that we have different types of workshops. We have Saturday workshops where everyone's there for pretty much 6 hours, 7 hours. You get fed really well, and then you learn how to teach one of our labs. Tomorrow we're having a Wolbachia lab. So we're going to be in the science garage at South San Francisco High School, and we're going to teach the teachers how to teach the Wolbachia lesson, as well as doing the hands-on. And then in the meantime, you get fed 2 great meals, and you also get some credit for CSU East Bay graduate credits. What we do is that we also provide equipment rentals. If you are in the Bay Area, then you can work with us to receive certain equipment that you might need for doing some biotechnology. We also have curricula, and here the curricula is actually free for teachers on our website, and I'll show you how to get into that later on. We also provide low-cost reagents. So here's an example of a bacterial transformation, protein purification outcome, where you see different colors, all from different bacteria that's been transformed with fluorescence.
You're muted, Yingsu.
How did that happen? So thank you for that. So you see that we are in the San Francisco Bay Area, 8 or 9 counties here, or not even 9 or 10 counties. And we go as far north as Calistoga and as far south as Morro Bay. However, we have expanded throughout the United States, and we now, since our partnership with InnovATEBIO, people know more about us through the summer series from 2 years ago when the pandemic really hit us. Okay, so this is just some data that's for you to see. We are in about 27% of Title I schools, high schools, and we reach over 200 teachers and over 20,000 students a year. So during the pandemic, it did go down a little bit, but since we had pivot to virtual lessons, teachers were able to use our lessons in their classroom, which I heard was really, really a great help to them, which is what we try to do. We want to be a great help and assistance to all our teachers. So where we feel that our strategic plan is in trying to create this biotech awareness that starts off middle school to high school and then going on to community college and beyond, right? And as we all know this, that exposure to biotech doesn't start early enough. And here is just a quote from a report that it's the lack of awareness and the lack of this pipeline. So our pipeline is there, but it's not big enough in order for students to be entering this biotech pathway. So that's where we try to be. We try to be at the 9th and 10th grade level, where we are introducing lessons to teachers and then so that they can teach their students and be much more aware of biotechnology. In our something new this year for us is that we are introducing a what is biotechnology lesson, working with new schools that want to work with us to introduce what is biotechnology using several different types of examples in a slide deck. And then the students have worksheets that they can work on. And then we also incorporate a hands-on lesson. It could be something as simple as micropipetting, but it could be something as complicated as PCR. And then all the way to having the students explore careers in biotechnology using biotechcareers.org. And then also exploring community colleges. So for example, we have 3 community colleges on our lessons, City College of San Francisco, Skyline, and also Laney colleges that we put onto our worksheets, and the students are exploring those biotechnology programs at the community college and seeing what's in store for them in the future, if that's something that they wish to choose. So something that we are doing, our strategy then is to do some teacher training via lessons and via workshops. We do it in multiple forms. It's not just a Saturday workshop. We can come to the school site, even if you're from another state. So we are starting to work with Nebraska and trying to do work there in terms of training. We can do it virtually, but we hope to visit Nebraska and they hope to visit here in San Francisco, so that we can give the teachers some hands-on training. And we're also working with the community colleges there so that we can help on this pathway from high school to the dual enrollment program that they have to the community college. So here we work with the teacher training at the high school. We provide the things that we talked about already: curricula, reagents, equipment lending. And we do this again by incorporating these pedagogical approaches. Something that we do is called Biotech Week, where we train the teachers in how to teach the lesson, and then we also avail ourselves to be in the classroom with them as they're teaching. So they have sort of a crutch, right? The idea is that we want to make sure that the 9th and 10th grade teachers who aren't very confident in teaching biotech, that we can help them set up if that's needed. We can help them answer questions. We are there to help them in the classroom when they need us. And we also can partner with industry for guest speakers if needed. So this is just an example of what happens. The whole class in 9th and 10th grade biology gets a chance at some hands-on biology. Okay, so I'm going to end here because this is about PCR. And then, so I'm going to show you then our website. Oh, I shouldn't have done that. Okay. Do this. Okay, you can see the slide deck, right? I'm going to show you our website. Can you see our website? Okay, great. So this is our website, babec.org. And if you're a teacher, you can now sign up to get into this website. We do have a wall, if I know that's what my staff calls it, in order to get in. I'll show you just around what we have here. We have what we do in history and our team. We also have some new initiatives that you can read up upon. And I see some of our partners are here, like Lori. She works with us in this BioScope project. And then we have some impact. But this is probably what you're interested in, the left side part of this website. So I will tease you with the other parts first. So we have some reagents here that you can order, and you can see what's in every one of these reagents by just clicking on these tabs. Okay. And then if you go to lab equipment, if you are lucky enough to be in the Bay Area, you can borrow some equipment from us. Okay. And you can see what's in each one of our equipment kits. And we've really streamlined it so that there's no extraneous things, because those kits do come back to us. And so it's items that you need for biotech. And it's not anything extraneous in the sense that you don't have to count all these different items, which can be painful. We've done that before. And then we also have professional development, of course. But I just want to take you to our curricula page, because this is really, we're really proud of our curricula. You know, we've been around for 25 years, and it's only been recently with our BABEC 2.0 team that we've gotten really good curricula writers who are teachers with credentials and with masters in curriculum development. And so we've worked really hard in trying to create lessons that are easily implementable by the teachers. And remember, the curricula is all free. You can access it as you wish. Okay, so I just want to show you some things to point out to you. You can click on this YouTube video. One of my staff, Michael, was telling me yesterday that I can go through the list of YouTube videos all weekend and really learn more about BABEC and what we do. I can also subscribe and like them. So we do have some COVID lessons in our midst. So it looks like the animation doesn't, oh yeah, here it is. So I'm gonna go back to that. We have some pretty cool animation if you're able to mouse over it. The pandemic project is virtual. It's a lesson that is having students looking at data, what is real data, what is fake data, and making some kind of claim looking at the data. So it's a really nicely done, I must say so myself, because I have some great staff on the pandemic project. We are still working on the COVID-19 testing lab. We're almost there. We do have a mock COVID kit that is aimed at community college teachers. If you're interested in that, let me know. We still have some to give away. But these are our usual labs that we have: micropipetting, electrophoresis, forensics, and then bacterial transformation. And then this is what we're interested in today, right? Because it's all about PCR. So we have these 4 PCR lessons. The ALU PV92, that's adapted from DNA Learning Center. Mitochondrial DNA PCR is also adapted from DNA Learning Center. And this one's really interesting because our lesson is, the question is there a genetic basis to race? And so it's a very appropriate lesson, especially after what the whole nation is going through right now with racial justice. So it's a very interesting lesson here. And we also have sequencing that can be done from the student's own DNA that they extract and PCR. Wolbachia is about this microbe that lives in insects. And maybe after this showcase, you'll hear more about how certain ways that scientists are using other microbes to help with the spread of mosquitoes and diseases. Wolbachia is one of them. And then our last one is GMO PCR, which I really want to change the name to GE PCR. And it's looking at food that students can collect. And then we extract the DNA out of that. And then we can amplify the DNA to see if there's any, if they have ingredients that are genetically modified in it. So I'm going to click on one of these just to show you what it looks like. I'm going to click on Wolbachia. And Wolbachia is actually adapted from our friends at Vanderbilt University. They have also a lesson on Wolbachia. So this is what you'll see then. It's an overview as well as learning objectives and prior learning knowledge. For high school teachers, we really strive towards alignment with NGSS, so the Next Gen Science Standards. I know not all states have adapted it, but California has adapted it in a huge way. And so when teachers are teaching this in your classroom, they really have to align their lessons with NGSS, and that's what we try to do. If you click on, I'm going to go to online lessons first. Oh, there is no 100%. Okay, sorry about that. So I'm going to click on the hands-on lessons, and this is what you'll see. You will see a bunch of little documents, right? And you can click on any one of them. They're all on Google, on a Google Doc. So let's look at the lesson plan. And I'm sorry about this, but it takes a little bit of time to load. Okay. So what it takes, what you see here is that it is a lesson plan that takes you through how you could be teaching this lesson in the classroom. And it even gives you time. It's going to take you 30 minutes to do this or 5 minutes to close. Okay. So that's what the teachers will be using. For the student, this is what they'll see. And since it is a Google Doc, which means that you can download it and then re-upload it to your Google Classroom or Google, Drive, and you can actually change it yourself. Okay, so it is customizable. We just ask that you don't sell the product after you customize it to your classroom. So this is a worksheet that a student can fill out that follows the slide deck that I'll show you next. So this is the slides that come with the lesson. And you'll see that it's phenomenon-based. If you're NGSS, teaching NGSS methods, then it takes you through these type of questions that you could be showing your students and having them come up with the questions to ask. What do you see? What do you wonder about? And so it's very rich in that area. There's also background readings that you see here. And if your students are EL students, we also have vocabulary tools where there's pictures as well as definitions. And then it helps the student really learn about how to use the language that we are teaching them in this lesson. So this is all what the teacher can use. So when it comes to the labs, we want to make sure that the teachers are empowered to do the labs themselves, right? After the first year, they may need help from us. The second year, they may need help, but just a little bit. By the third year, we're hoping, hey, you can do this on your own now, right? And how do we do that? We do this by having these really important preparing the classroom. And, if you're Bio-Rad, you'll say, okay, that's very similar, right? Bio-Rad has this too. So we have, a way to prepare all your solutions, how to aliquot it, so that you're not aliquoting for each student, but how to do it in a group of 4. So it's saving you time and the calculations involved. And then we also have how to set up what your groups can look, what your group setups can look like. You'll need, blue tips, yellow tips, the 3 sizes of micropipettes, as well as, all the different tubes, et cetera, et cetera. And this, of course, is going to relate to the reagent kits that we give out. Okay. We also have then an inventory guide. If you buy a PCR reagent kit from us, this is what you'll get. Making sure that you have these reagents that you requested. And then you also need these other things, right, that you may not have. And that's very similar to, I think, to what Bio-Rad has. And then finally, what we have too is a visual way of doing the lab. And this is very helpful for students because you almost don't even need to read it in order to do this lab. So you can see that, you have an insect and you're grabbing the insect, and you also need to measure the insect and what it looks like, and where to put it into a tube, and to use a fresh pipette tip every time, how to grind it up. And we try to have this in such a way that we're having students, so this is an actual student from a teacher, come and help us with this lesson, so that we were able to take pictures of them. So it's a very well supported protocol. The protocols themselves, you cannot change, although, because we don't want you to change them, because it might not mean that you'll have success. We want to make sure that you follow our protocol so that you have better success. And then you have the supplemental resources, anything that you might want to give to your students for extras. I want to show you something that we have put in here. It's the What is PCR? It's an activity guide that goes with a video. So you might be teaching PCR for the first time, and you want to make sure that your students are seeing it, and you'll see that it's a video from DNA Learning Center. Okay, and you can click on that. It'll take you to a YouTube that shows you what PCR is. And it's just an introductory lesson for the students to first learn about PCR. And then they can answer questions and get them introduced to the concept before they actually start doing the hands-on. So it could be a flipped classroom place where they can do this for homework, or, if you, God forbid, we go back to doing things virtually, it could be something they do at home. Okay, so I think that's my spiel. And I take any questions, and I'll put my email on the chat.
Nice job. Thank you very much. Very impressed. I'm kind of curious. So now that you're working with Nebraska, are you willing to work with other states and individuals?
We are, that's a good question. So BABEC, we're entering BABEC version 3.0 is what we're calling ourselves. Since I took over in July of this year, our team and I have really are trying to operationalize a lot of our processes that for the last 25 years has not been operationalized. And because of that, we are expanding. And so if you would like to work with us, let us know. We can be on grants together. For example, with Nebraska, that's how it's working. We are working with them through InnovATEBIO, but they also put us onto their next ATE grant, which they will submit by October 14th, and I hope they get. So we can work on grants together if that's something that you want us to do. Right now, we're working with quite a few school districts in California, something called the Strong Workforce Program, and we are fully in almost a huge part of the 9th grade biology classes in San Francisco Unified. We are also with the County Office of Education with Contra Costa County. And we are in most of the districts in the peninsula area. So, that's been really the best thing for us, because we love working with and trying to get into the classrooms in each one of those biology classrooms so that all students have access and not just a few. And then so I think that by widening that pipeline, we'll hopefully get more students to be more aware of biotech so that we can fill the gap for the workforce. So yes, the answer is yes. We can go outside of California.
You should do a collaborative grant next year for $3 million.
Okay. I'll put that on my list.
Good.
We do
Yeah,
want to do
so
an
easy.
ATE grant. So.
Well, thank you.
You're welcome.
And this was so informative. Thank you so very much. Absolutely.
Bye-bye, guys.
Yeah.
Bye.
So did you, Lee, did you have anything else to add?
No, but I'll play the GCTA song if anyone wants to stick around for it. You don't have to, of course. It's Friday afternoon. Go enjoy your weekend. But it is really funny. So I'm going to share the screen and y'all leave whenever you want.
Okay.
Oh, that'll be fun. Thank you very much. And
Thank you.
please please everybody take the survey if you haven't. If not, we'll haunt you with it with emails. Thank you.
Okay.
All right, here we go.